Chondrocladia lyra, also known as the lyre sponge or harp sponge, is a species of carnivorous deep-sea sponge. Its domain is Eukarya, its phylum is Porphyria, the class is Demospongi, its order is Poecilosclerida, the family is Cladorhiricidae, the gender is Chondrocladia, and its species is lyra. According to the article The Systematics of Carnivorous Sponge and the research that they did with DNA of some carnivorous sponge, as you see in the phylogeny, in our molecular analysis, carnivorous sponge were recovered as a monophyletic assemblage closely associated with Mycalidae and the rest of Guitaridae. However, the analyses were not available to completely resolve whether Guitaridae or Mycalidae is the closest sister group of if they constitute their own separate clade. The original name of the organisms that we are learning is Chondrocladia lyra. This is the same taxonomic name. The type of location for this species is of California at the South Scavanna Rift, with additional reported sightings at the Monterey Canyon and a deep range of 3,300 meters to 3,500 meters. About the geographical distribution, the first discovery was in the California coast, living at depths of 10,800 to 11,500 feet by the Monterey Bay Aquarium Research Institute. In this image, you can see the distribution correctly. We found that animal in the northeast Pacific sides of Scanaba Ridge and Monterey Canyon. Then we're going to talk about the environmental niche. It's a marine animal who lives without light. But how they can reproduce? They have a structure that is made up of horizontal branches. They leave vertical trees that end in species of balls. Biologists believe that these structures are used for reproduction since they contain sperm. A symbiosis relationship with other organisms is not possible because the environment where it is found is so hostile and in the midst of such extreme conditions that very few organisms can survive in this kind of environment. In my point of view, the public should know about these animals. It's an avid carnivore that relies on water current to push tiny crustaceans into its picket fence-like upright branches. You will see these branches have tiny hair-like structures called filaments splitting off horizontally. Each of these is possibly packed with tiny hooks known as spicules, perhaps 5,000 on each filament, which act essentially like Velcro to snare minuscule prey. As the catch struggles to free itself over the course of 10 to 44 hours, the cells on the sponge branch crawl up onto the critter. There's no beating and there are no toxins involved. The prey is simply immobilized and subsumed right into the sponge. And then once the prey dies in there, the cells around it migrate into any little opening through the mouth, through the nose, through whatever openings there are between the skeleton, and starts to dismantle the cells of the small crustaceans. Finally, the phylogenetic tree was made with the species provided, including the one studied through this video. This tree was made taking into account the taxonomy of each species, starting with the domain, continuing with the phylum, and up to the class. In the images, we can see some of the species that were used to make the phylogenetic tree. Finally, different sources were seen that provide the information in this video in addition to previous knowledge through the course of organisms biology. Thanks for your attention.